Okay, so we are on part 6. Part 6 is going to be about DNA. So now we're talking more towards the larger aspect of what biology is about. So DNA, genetic material, going all the way through evolution, how we evolved from something so, how life itself evolved from something so small to something so large. Uh, but DNA is that building block that we have to go over. So DNA, or deoxyribonucleic acid. This is your long molecule that contains all the information to make a copy of an organism. DNA has 3 major parts: a phosphate group, a nitrogen base, and a sugar. So when these things are combined, you create a nucleotide, a nucleotide, a nucleotide, either one. Nitrogen-containing base consists of 4 different types of bases. You have adenine, guanine, cytosine, and thymine. They are basically 2 different types. So you have your 2 rings of carbon and your single ring of carbon. Your purines are going to be your 2 rings, and your pyrimidines are going to be your 1 ring. DNA is always in the shape of a double helix. So when we say double helix, it kind of looks like a twisted ladder, and that's how it's connected. It was discovered in 1953. The rules are that complementarily based pairings. That means rules tell you that which nitrogen base will always go with the other. Rule number 1, A always goes with T. So adenine with thymine, they will form 2 hydrogen bonds. Rule number 2, cytosine will only combine with guanine. So they have 3 hydrogen bonds. So the replication process of DNA in a cell is called meiosis. That is the start of it. So in meiosis, we have the nucleotide chain separates. So now we have 2 different pairings. They have replicated into a fork, so replication fork. The chain separates into enzymes called helices. These break down hydrogen bonds between nitrogen base. So step 2, DNA bonds to DNA. So the new strands of DNA are made up of nucleotides. They will now create new nucleotides. So they have now duplicated. And of course, A with T, C with G. The DNA polymerase continues until it reaches the end of the DNA strand, or another molecule. Replication is now over. So now the replication is complete. You have 2 exact copies of the same DNA. So DNA replicates with very high accuracy. There's only an error about 1 in every 2,000, 10,000. So that is called a mutation. If an error occurs in a gene, serious effects can result. You can look at the different things once you get to anatomy or something like that. They'll go over basically what these mutations are. DNA enzymes to proofread the strain to correct the errors if any harm is done. So transcription involves RNA. So there are 3 types of RNA. Messenger RNA, which carries the genetic information from the DNA to the ribosomal cell, to the ribosomes. Transfer is a single chain that attaches to the messenger, and it basically moves this towards the ribosomes. And ribosomal RNA, these are the most abundant. Their function is to create protein, so protein synthesis. The process of transcription carries the information of the DNA to the ribosomes, to the cell outside the nucleus, to form messenger RNA. So RNA polymerase makes copies of specific sequences in the DNA. It binds with specific regions of DNA called promoters, which in the beginning of DNA will be transcribed. So the transcription, transcribe. Transcribe is basically writing it down. Step 2, RNA polymerase continues adding complementary nucleotides to make the mRNA. Nucleotides are added until the RNA polymerase reaches the termination state signal. So, specific sequence of DNA that makes this the end of the gene. So DNA has a sequence in order to know what ends the gene. RNA polymerase then releases the DNA, and newly formed messenger RNA then leave the nucleus. The three types of RNA made in this manner. So the products of transcription are three types of RNA. All of them are involved with making protein. So protein synthesis is called translation. So transcription, copying down all the DNA sequences and genes. Translation is how we turn that, basically thinking of how we translate that transcription. This is the process of how to piece together the genetic information. So proteins are made up of small amino acids. There are 20 different types of amino acids. These are all linked together to form protein itself. Genetic information is needed to make the proteins. That's why you need a series of messenger RNA. A few amino acids don't count for any code. These are your amino acids that don't count for much code. These stop. Transportation RNA is also known as an anticodon or codon, while the three base pairs of mRNA are known as codon. So remember, proteins are made up of three-dimensional structure called amino acids, just like DNA is made up of the double helix. The first amino acids can arrange the chain sequence. This sequence then determines how the molecule twists and folds upon itself. This twisting and folding determines the protein's three-dimensional structure. So these structure will be different based on the type of protein they are. There are 20 different types of amino acids. So genetic code is what is in our DNA and is transferred on messenger RNA. The set of three messenger RNA nucleotides are called a codon. Each codon has codes for a specific amino acid. And don't worry about the book. So if you need this, I will give it to you, so don't worry about it. So all organisms use the same code to do this. So no matter if it is a plant, animal, or human, they all use the same thing. Because of this, it supports the theory that organisms are evolutionarily related. That's why there is a specific parts of the DNA that are the same in all organisms on Earth. Certain sequence of the messenger RNA don't code for the amino acid. They code for the start and stop area on the messenger RNA. So this codon tells the ribosomes to start translating to the messenger RNA into protein. The stop codons are the UAA, UAG, and UGA. Most often the UAG is the codon that stops, but that is possible for the other two to be the stoppers too. The process of translation. This process starts as messenger RNA that left the nucleus and reaches the ribosome. Number two, amino acids then are floating freely through the cytoplasm, and they will be attached to the transportation RNA. Amino acid is attached to the opposite side of the three-base nucleotide sequence. This sequence attaches to the messenger RNA, and it is called an anticodon. As more and more transportation RNAs are brought into the ribosome amino acids, they are bound together into a peptide bond. So this continues, the ribosome move along the messenger RNA until the stop is reached. So the protein folds upon itself to create a three-dimensional structure, and the protein is made. Proteins are used to bind the entire structure together and make sure that your entire cell works. Make sense? All right. That is all for today. Go ahead and take the quiz that is attached. You need to get 7 out of 10. I believe there are 12 questions this time, so you need to get, so you'll even be lucky enough to miss. You need to get 8 out of 12. All right. Good luck.